In the black water below the sunlit ocean, pale fragments drift downward, like a storm moving in permanent night. This is marine snow, a loose procession of dead cells, waste, mucus, molts, minerals, and living microbes. If you follow one flake, its descent becomes a contest between gravity and countless hungry mouths. The particle may begin as phytoplankton, dividing near the surface where light and nutrients permit growth. A grazer bites the cell, releases scraps, and packages other remains into a compact fecal pellet. Sticky gels collect fragments, and collisions build aggregates large enough to sink beyond the illuminated layer. Below, no green plant can replace that food through ordinary photosynthesis because usable sunlight has vanished. Yet bacteria colonize the falling material. Zooplankton intercept it, and seafloor animals wait beneath its path. The mystery is not whether food descends, because sinking particles are directly collected and observed. The mystery is how much survives, who consumes it, and which events deliver enough to sustain deep communities. Most organic matter is altered or eaten before it reaches great depth. Every bite transfers energy into a body, while respiration returns part of its carbon to surrounding water. The snow, therefore, thins during descent, feeding a vertical ecosystem by disappearing into it. Its apparent gentleness conceals rapid recycling, selective feeding, and occasional pulses of extraordinary abundance. The investigation follows carbon from a bright surface cell toward a seabed that may lie kilometers below. Marine snow forms through biological production, particle collision, mucus secretion, and the attachment of mineral grains. Individual pieces range from barely visible specks to large, fragile aggregates with intricate porous structures. Their sinking speed depends on size, density, shape, water motion, and how organisms modify them. Dense fecal pellets can descend quickly, while fluffy aggregates may hover, fragment, or be carried sideways. Transparent exopolymer particles provide sticky material that helps small cells and debris combine. Larvaceans filter tiny particles through mucus houses and discard those structures when filters clog. The abandoned houses sink, carrying captured material into deeper water as unusually large packages. Salps also filter surface particles and release fecal pellets that can export carbon rapidly under suitable conditions. These examples show animals creating marine snow rather than merely waiting to consume it. Once a particle enters dim water, bacteria attach and release enzymes that break complex material into absorbable compounds. Their growth attracts protozoans and small animals, turning each aggregate into a temporary drifting habitat. Copepods detect chemical or mechanical cues, approach particles, feed, and sometimes fragment them into slower pieces. If you watched only the largest flakes, that fragmentation could make export appear to vanish without revealing its consumers. Some zooplankton migrate upward at night, feed near the surface, and produce waste after returning to depth. This active transport delivers material below without requiring every carbon atom to sink as passive snow. The mesopelagic zone becomes a processing layer where particles, migrants, microbes, and predators repeatedly exchange energy. Oxygen is usually consumed there as organic matter is respired. helping shape midwater oxygen distributions. Below roughly 1,000 meters, particle supply is smaller, but long-lived animals have lower metabolic demands. Many deep species grow slowly, conserve energy, and respond strongly when rare food pulses arrive. On the seafloor, brittle stars, sea cucumbers, polychaetes, amphipods, and microbes consume settled organic material. Deposit feeders ingest sediment, digest usable compounds, and leave reworked grains behind. Suspension feeders extend structures into currents, capturing particles before those particles touch bottom. These documented pathways establish marine snow as a major connection between surface production and dark ocean food webs. The first uncertainty concerns quantity, because collecting a fragile, moving flux without disturbing it is difficult. Sediment traps intercept falling material, but currents, swimmers, and hydrodynamic effects can bias what enters. Optical cameras count and size particles through limited volumes, then estimate flux from assumptions about sinking speed. Stable isotope measurements trace dietary sources, although overlapping signatures can prevent exact identification. Incubations measure microbial processing under controlled conditions that cannot reproduce every turbulence and pressure regime. Together, these four approaches constrain the journey while exposing different blind spots. The next uncertainty concerns quality, because equal masses of snow can offer very different nutritional value. Fresh phytoplankton-rich aggregates contain more accessible compounds than particles repeatedly consumed and repackaged during descent. Mineral ballast may increase sinking speed, but minerals also add mass that is not edible organic carbon. Fast descent can preserve energy by shortening exposure to consumers. Although speed varies during the journey, the ballast explanation is mainstream but not universal, because mineral type and aggregate structure change outcomes. Another hypothesis proposes that microbial degradation changes aggregate structure enough to slow or accelerate sinking. Such processes may matter in particular aggregates, but their global importance remains uncertain. Direct observations show fragmentation and consumption, while many encounter rates are inferred from particle changes across depth. An animal beside a particle is not proof of feeding unless behavior or ingested material confirms the interaction. Likewise, a sudden seafloor deposit does not reveal whether material fell vertically or arrived through lateral transport. Underwater slopes and currents can move organic matter far from the surface waters where it originated. Seasonal blooms offer a stronger sequence because surface production can be followed by increased particle export below. At some sites, a later pulse reaches the seabed and triggers rapid feeding, reproduction, or movement. The delay varies with depth, particle speed, consumption, and horizontal advection. Not every bloom produces a matching deep pulse because surface grazers may recycle most material before it sinks. Conversely, storms can inject particles downward or resuspend sediments without a preceding local bloom. These mismatches prevent simple one-to-one links between a satellite visible bloom and a deep feeding event. Large carcasses form a separate category of food fall, far rarer and richer than ordinary marine snow. They create intense local communities. but they should not be mistaken for the continuous background particle flux. Chemosynthesis supports additional deep ecosystems at vents and seeps, making sunlight-derived snow dominant without being exclusive. Unknowns persist because small particles and dissolved compounds can feed microbes while escaping visual definitions of snow. The journey matters to climate because sinking organic carbon can be isolated from the atmosphere for varying periods. Carbon respired just below the surface may return to the air relatively soon through circulation. Carbon carried into deep water can remain separated much longer, depending on where currents eventually transport it. If you change particle size or sinking speed, the depth of remineralization can shift even without changing surface production. This sensitivity makes the biological carbon pump responsive to food webs, mineral inputs, and physical mixing. Warming may alter phytoplankton communities, changing cell sizes and the kinds of aggregates they form. Stronger stratification can reduce nutrient delivery to surface waters, potentially lowering export in some regions. Ocean acidification may affect organisms that produce calcium carbonate, with consequences for particles and ballast that remain regionally complex. Declining oxygen can change which midwater consumers survive and how efficiently they process sinking matter. Fishing can remove animals that package, transport, or consume carbon, although ecosystem-wide consequences are difficult to quantify. The deep seafloor experiences these changes remotely, receiving altered food from events far overhead. A sea cucumber encounters a fresh deposit, turns toward it, ingests sediment, and concentrates scattered nutrition inside its gut. This Q-action adaptation sequence converts a brief pulse into stored energy for maintenance and reproduction. Some abyssal animals produce young or increase activity after food arrives, linking life cycles to distant surface seasons. Others remain metabolically subdued, an adaptation to long intervals between meaningful deliveries. Microbes respond within hours or days, using compounds that larger animals cannot access directly. Their respiration releases nutrients and carbon dioxide, continuing the recycling that began near the surface. Burrowing animals mix oxygen into sediment, changing where microbes can operate and which chemical reactions dominate. Only a small fraction of surface organic production is ultimately buried, but that fraction influences long-term carbon cycling. Marine snow, therefore, feeds organisms, shapes oxygen demand, and connects climate processes with hidden animal behavior. Future evidence must resolve individual particles without losing the regional and seasonal scale of export. Long-duration cameras can record pulse timing, aggregation, and fragmentation through changing conditions. Improved traps can better exclude active swimmers and compare collection under different current speeds. Stable isotope patterns can test how particle sources change while material descends through microbial processing zones. Pressure-aware experiments may reveal whether deep microbes process fresh material differently from decompressed laboratory samples. Linked surface and seafloor observations can test which blooms actually generate food pulses at depth. Measurements across oxygen minimum zones can show how consumer loss changes particle survival and remineralization depth. If you meet the original pale fragment again, it will probably no longer be the same object. Microbes will have colonized it, grazers may have bitten it, and turbulence may have split it apart. Some carbon will enter swimming bodies, some will dissolve, and some will continue toward the bottom. The fading particle is not failed delivery because its disappearance records the feeding of mid-water life. What reaches sediment becomes another beginning, awakening consumers and microbes in a world without sunlight. What never arrives has already supported organisms suspended above that distant floor. The final accounting remains incomplete because fragile particles transform faster than any method can follow every component. Marine snow sustains the dark ocean through a diminishing, changing flux whose pathways are observed, interpreted, and still only partly measured.